Hello, I hope you are, can you hear me? Okay, I guess you can. Oh my God. I'm listening myself. I'm listening to myself. Oh, and delay. Yes, I know. Okay, now. Okay, I'm very sorry. I didn't hear the presentation of Arne. I'm sure it was okay. I first of all wanted to thank the organizers for inviting me to this Congress and to express that I am very sorry that eventually I could not come. I would have loved to be in Brazil right now and see all the presentations of everybody and interact with all of you. Having said that, I'm going to start. As you know, the 2030 Agenda for Sustainable Development of the United Nations demands, among other things, to assure food supply via the implementation of sustainable and resilient agricultural practices that increase productivity, improve land and soil capacity, and strengthen agricultural systems' capacity for adaptation to climate change. Biological control is at present one of the most sustainable and resilient agricultural practices because it relies on the very same mechanism upon which herbivore populations are regulated in nature. That is, the secondary consumers feed on primary consumers, and here we will find the pest, to induce an indirect effect to trophic levels below that we call trophic cascade, and that results in an increase in the primary producers, the plants or the crops. Furthermore, it complies with all the pertinent regulations, the current regulations from Europe and from the world that are basically aimed at reducing the use of pesticides or to promote the use of autochthon species for biological pest control. But unfortunately, and according to the literature, I want to stress that, it's not that I get it from my pocket, this is asseveration. According to the literature, biological pest control has quite a low rate of success. Most of the cases of unsuccessful biological control comes from the failure of natural enemies to establish in the crops. We believe that this low rate of success is caused by several factors. One of them is that biological control assumes that each predator will fit in each pest, all of them interacting, forming trophic change, when we all know that the species that are inhabiting crops interact with each other, forming sometimes quite complex food webs. They also ignore the effects of abiotic factors on the dynamics of communities. And because these two factors are strong selection pressures, they ignore the evolutionary processes that likely are occurring in the field. This is why some time ago, a colleague of mine, we came with food web engineering, that we define it as an extension of biological pest control that integrates community ecology and evolutionary sciences into the management of agricultural systems potentially exposed to hostile environments. Basically, what food web engineering pursues is to exploit the inherent intraspecific genetic variability of all biological control agents. This is and is done using artificial selection when the trait that we have to improve is a priori known, or experimental evolution when the trait or traits that should be improved are a priori unknown, with the ultimate goal of going faster than natural selection. This is the scenario that I will stress during this presentation. It's a scenario of interest in which BCAs are exposed to their own predators and exposed to warming. The literature provides a comprehensive list of traits that are amenable to be artificially selected. This is true, but the problem that we have is that under a scenario, a specific hostile scenario, many times we don't know what trait we should improve. But what do we know is that these traits should be response traits. This is traits that hold enough genetic variability to be able to respond to perturbations. And furthermore, we also know that these response traits should also be effect traits. This is traits that affect the properties, functions, or services of ecosystems. In our case, what we are interested in is in the strength of trophic cascades. So what food web engineering proposes is, first of all, to know the genetic architecture of the traits, then to cross it with the several communities, several configurations of communities that we know that happen in the field, and different scenarios or different abiotic conditions. Then to use this data to feed a next generation individual-based model that I will tell you about it later. Next generation's IBMs are IBMs, individual-based models, that also allow to include normal differential equations or different equations, the equations that we normally know. And once the simulations will tell us what rate of combination of traits would maximize our trophic cascade. The next step would be to apply experimental evolution studies and to test the newest strains that you get in the field. And continue the circle is if needed to get more realistic data. Or in other words, what we propose is to first ask, are they response traits? What of the traits are response traits? Which ones of these response traits are also effect traits? And do the genetically improved strains work better in the field? So what I'm going to explain in this part of the talk is this first box of the schema that I just showed you, that answers what traits are response traits. And this was done by a former PhD student of mine, Diego Serrano Carnero, that got his PhD position thanks to this trait. He defended this project. He defended his thesis in December 2023. So the first thing that Diego did, oh no, sorry. To do this work, we use as experimental model Amblyseius swiski, because it's one of the species, if not is the species that at present is most used in as a biological control agent, because it's quite good fighting against 3 of the 4 super pests, that's spider mites, whiteflies, and thrips. The 4 super pests would be aphids, and it has a very short generation time that makes the species very amenable for evolutionary studies. So the first thing that Diego did was to travel to Israel to get and sample different crops in different locations to get a wild population of Swisky, because this species, Amblyseius swisky, is originary from the Middle East. And he came back with more than 500 individuals, and we cultured in the lab 2 populations, the wild population and the commercial population. The next that we did was to create isofemale lines with the goal of distributing all the phenotypic variability of the population, the whole population, into subpopulations that are created from one single female, with the aim of increasing the genetic variability, the variance between isolines, while decreasing the variability within each isoline. And once we got, with this process, we managed to obtain 20 isolines from the wild population and 22 from the commercial population. The commercial population, we got it from a company. And after the next step that we did was to measure a total of 23 traits on 18 of them on the same individuals. These traits, we categorize it into different categories: life history traits, behavioral traits, and morphological traits. We also measured several physiological traits in different individuals, because measurement implied either the death of the individuals or a change in the diet. And this is how we got the genetic architecture of traits of Amblyseius swisky. In here, you can see the significant genetic correlations between traits within the same category and between categories. All the genetic, the significant, both positive and negative genetic correlations. To reduce the number of traits to continue with the process, we applied PCA. But in this slide, you can already see that all the multidimensional variance of the commercial population is contained inside or within the variance of the wild population, both for females and males. The next thing was to estimate the heritability using the intra-class correlation, which is the proper way to estimate heritabilities when you deal with isofemale lines, because David et al., with this formula, it calculates it, but correcting by the number of isofemale lines. And indeed, we found heritability in traits related to size, development, reproduction, and physiology. But one result that claimed immediately our attention was that none of the traits that were measured in the commercial population were held an heritability significantly different from 0, which is not good, because this indicates that probably when these individuals that are released in the field have the capacity to adapt to different environments quite limited. Probably is quite limited. Very well. Once we got the potential response traits, what we did is to check whether they are able to evolve and towards what direction. For that, as I told you before, we tested this scenario in particular, in which the natural enemy is confronted against his own predator in a biotic selection force, and to high temperatures in a abiotic selection force. The experimental design, you can see it in here, is a full two-factorial design with presence or absence of top predator and mild or hot temperatures. We applied experimental evolution for 10 generations. After that, we put all the populations in a common garden with very nice conditions, and to eliminate the maternal and the grandmaternal effects. And after that, we started measuring the traits. And indeed, we found evolution in traits related to size and development. Indeed, the size of the eggs in blue, in light blue, it's each of the replicas. You have to be aware that our unit or replic unit is the population, not the individuals. So you have 6 replica, and in dark blue is the average of all the replicas. So what we found is that the eggs became smaller at higher temperatures, but only in the presence of their own predator, the integral predator. Both the females, both the eggs that gave females and or that gave males. All the eggs were smaller. And the same happened with the size of the adults. The females became smaller when they were, for 10 generations, at high temperatures and in the presence of their own integral predator. As for the development, individuals, it took longer for individuals to develop at high temperatures and independently of the presence of the integral predator. So basically, we got results that are partially, at least, that are disagreeing with the temperature size rule in ectotherms. These rules, basically, what it says is that individuals that are exposed to high temperatures will develop faster, but at the cost of becoming smaller. That rule was described at Hutchinson in 1994, and from this time there has been several considerable number of experimental cases that are exceptions to this rule. One of them even from the same guy that made this rule. Then later on, some papers appeared as kind of trying to describe the conditions upon which these rules should actually happen. And the last one is they're saying that there is actually no evidence that warmer temperatures are associated with selection for smaller body sizes. But don't forget that we found that we got that the populations evolved towards the smaller sizes when temperatures were higher, but only when the predator was present, their own predator was present. To know the genetic architecture of traits may contribute to explain the results that you have of the results of evolution experiments. For example, in here, these negative genetic correlations tell us that the more active an individual will be, the smaller the eggs this individual will produce. And in here, these positive genetic correlations indicates that the smaller are the eggs, the smaller will be the adults. So from this, we made our hypothesis of what factors could have favored individuals to become smaller. For one side, we have this guy that at higher temperatures, it will move much more. It will produce smaller eggs that will result in smaller adults. And in the other side, we have this predator, Orius laevigatus, which is a visual predator that if it forages according to the optimal foraging strategy, is probable that he will first hunt the bigger individuals. Furthermore, An increase at higher temperatures will increase his activity and probably will increase his predation rates. All of this favoring for individuals to become smaller. With regard to the developmental time, being true that increasing temperatures increases metabolism in ectothermic species, it is if we separate catabolism that relies on chemical processes and is very depending on temperature, and anabolism, which only relies in physical processes and are quite independent to temperature, if individuals to move from one stage to the other need to fill a tank of energy before passing from one stage to the other, it is very probable that because at higher temperatures you also need more energy for maintenance, altogether can delay or make longer the developmental time. I cannot see you, I cannot see your reactions, so I hope that you are understanding what I'm saying, otherwise you ask it in the end. But it's very clear. If you need to feel this, and before you reach the top to pass here, if you spend more in maintaining, this will delay your development. The next step, as I told you, was to see what is the part in which we are interested in knowing what of the response traits are also effect traits. For that, because to do experiments to test that would be impossible, we use modeling. And we got actually this project, Jordi and I, that is specifically directed to this modeling. What we are what this guy over here, David Pablo Quevedo, is doing is parameterizing. This project is still ongoing, so we still don't have final results. But what it's doing is to parameterize multiple biological control systems with our own data from the literature, run simulations under different scenarios, and we expect that the outputs would tell us what combination of parameters make crop more persistent, or what combination of taxa, what functional traits, or what functional types or traits should be more appropriate to keep pest species at low numbers under each specific scenario. The guy that has done this program, this model, is Jordi Moyo. So I will later explain you a little bit about Weber, but if you want to go deeper to the details, you should contact Jordi. As I said, Weber is a next generation individual-based model that allows for different degrees of connectance between species. And it allows also, you can incorporate different space heterogeneity using cells. In each cells, you can increase the distance between cells. And it allows for genetic variability in traits. Therefore, it allows for eco-evolution of traits. And furthermore, it can depict trophic configurations of specific agricultural communities, with the aim of offering solutions a la carte, if we manage eventually to do it. In here, now I'm gonna show you examples. This is one of the simulations of Weber in a system that is composed of a predator, a prey, alternative prey for the predator, and biofilm biomass. Biofilm biomass is the resource, the base resource. That would be the plant. And as you see, after many days, the system crashes and only the plant remains, which that would be good. But I want to, what I'm going to show you is what happens with only including genetic variability in one trait, the distance at which the prey can detect the presence. We pass from a system that crashes to a system that shows high persistence, that it persists through many times, a long time. And continuing with the examples, with regard to the space heterogeneity, I wanted to put videos, but okay, what we pretend to find out, including the space, the heterogeneity of the space, is to mimic greenhouses and what would happen inside the greenhouse. So, and then adding different things we will be able, or we are able to see what would happen in for the production of the crop or the biological control of pests. We can add different islands of resources. So island of resource, we understand from like a bunker plant. It's an island that you plant inside or outside the greenhouse that holds that there are predators inhabiting this island, and then they spill over to the greenhouse. And I'm going to show you some screenshots that I did from the videos. These are demos. These are not simulations, are demos. In here, for example, you have a system with the basal results and a prey. This is 2 greenhouses, although you only see like it is in one, but you will understand further why I'm saying that. And what happens, as you would expect, if you go from the left to the right, is that the pest colonizes all the plants, and eventually all the plants and the 2 greenhouses, and eventually all the plants are infested with the pest. In the second scenario, if we put 4 islands in the corners, it's highlands with plants that are hosting predators, and we run it, and we see that also is not a good result, but you will reach this phase much later than in the previous one, obviously. But eventually, the predators manage to pass to the next greenhouse. Now we try a hedgerow with several plants that host predators, and it's more or less the same, but it goes again slowly. It has improved the output or the control, although it's not that good. But if when under the last scenario, when we add the hedgerow and 4 plants in the corner, eventually the first result that you can see is that the pest never manages to pass to the other greenhouse, and eventually the predators control the pests. And how are we? I'm at this point of the talk, I think I'm going very fast. At this point of the talk, I wanted to show you some examples that we have been doing, so applying this a little bit in the real life. What we see here is what we call the plastic sea in Almeria, that can be seen from the International Spatial Station. Imagine, here is zooming up, and this is what you see. It's just the sea of greenhouses with plastic greenhouses. And it's located in the southeast of Spain. And in there is these 2 colleagues of mine, Monica Gonzalez and Estefania Rodriguez, that they have the aim of making making Almeria green again. And this is what they do. They start installing a lot of hedgerows between the greenhouses with plants, autochthon plants, that they know that hold the species of natural enemies. And also, they do the same in hedgerows or plants inside the greenhouses. They even take care of the lagartos. So people that don't disturb the lagartos, they also contribute to control, I guess. And to finalize, I think I've been quite fast, but anyway, it's good because we started late. I'm going to explain you the last, another applicable research that we did following this I cannot come with the word. In this project in here, we were asked to get strains of Swisky better adapted to low relative humidities. How did we know that we could do it, or could try it at least? Because we got heritability significantly different than 0 for these three traits. And furthermore, from the genetic architecture, all these positive correlations indicate us that an individual that is more resistant to a relative humidity of 60 very likely will be more resistant to lower relative humidities. So what we did was to expose populations, because there were quite a lot of individuals, 200x, to different relative humidities. In one was the control kept always at 75%, and the second one, it started at 65, and then we decrease depending on the results. What we got was this. The first round of selection, we already had a big decrease, but we managed to recover it. Then the populations struggled quite a lot at 60%, quite a lot during 5 rounds, until we got this peak, this increase of survivals, and we repeated at 60 again, and to see that whether this result was robust. After that, we have now a population in a standby to be exposed to 55% relative humidity, and this population, it was phenotype. The phenotyping was done after many generations in common gardens, so that these populations were not exposed to this selection pressure anymore. And after all these many generations, 2 of them still kept the resistance to low relative humidity. Be aware that 60%, I mean, this is 60%, but it's the real relative humidity. We know that in the surface of the leaves, normally it's higher than the ambient relative humidity, but in this is the real relative humidity. And we phenotype for different traits, and we did not find any relevant result, at least for the developmental time. So we tried it in 2 types of food sources, pollen and in thrips. The thrips that we used was Thrips parvis pinus, which is an invasive species. Here, for developmental time, no problem. For predation rates also, we didn't see any negative effect. So that selection did not affect negatively, at least in this trait. The same for oviposition rates. Even some lines had higher oviposition rate than the control. And just to finish, to tell you that these 2 populations or subpopulations were delivered to Agrovio, which is a company that massively produces natural enemies or beneficials, and it's a small, medium Spanish company. And I'm sorry, but I finished my talk. Maybe I was too fast. I don't know how long it took, but I wanted to just introduce you to the people, to my colleagues that participate in any way to these projects. This is David Pablo Quevedo, Jordi Boya Laraño from another research center belonging to the CSIC that is located in Almeria. Inés Fragat and Sara Magallanes from the University of Lisbon, and these 2 girls from Almeria, and then from the IHS Melamayora, we have Juanma, Magdi, Diego Serrano, Rocío, Camero, Efren Millet, Inma Torres, and myself. And thank you very much for your attention, and I'm going to stop sharing the screen.
Thank you, Marta. I don't know if you can hear me. Yes? Okay. So I want to open the presentation for questions from the audience. Any questions? No questions. So I guess it was very clear.
Was I too fast?
Yes. Ignore Angelo.
Angelo says yes, but Angelo. Madeline says no. You know Madeline. Trust her. Was it understandable?
Yes.
Yeah? Good. But there are really no questions? No questions, Martha. That's a bummer.
Oh my God. Anne. Anne, I'm sure you have a question. Anne, I don't hear you.
And me.
Oh,
Sam, I exist.
Sam, of course. Sam is here too. Um, the audience may not know, but Angelo and Madeleine and Sam spent time together with Martha in Amsterdam, also with me. So it's a group of old friends, and that's the more the pity that you couldn't be here. But we all hope that you will recover fast, and there will be other opportunities. Thank you very much again. And that closes this session, which means that we will have time for all the slides of Sam, I think. Thank you very much. Thank you all.